In this session, we will learn how to use Blastweb, a publicly available resource, to find related protein sequences. In order to do this, it will be useful to have the refseq protein ID handy for the protein that you're working on. In our example, of course, we will use the CFTR protein. And then you can navigate to the blast.ncbi.nlm.nih.gov site. So when we go to the BLAST website, let's start with the Protein BLAST section. So we'll click that, then we'll type in the reference seq for our protein, NP_000483.3. Then we also want to make sure that we've selected the refseq option for running our BLAST. It's defaulted here, but you just want to verify that that's what you'll be using. And then click BLAST. Now this can take some time, so I've cut out a part of the video with that, and we'll just fast forward to the end results. So in the results, there are a few things that we can see. Among the first parts of the information is the ability to look at protein motifs that are commonly found in animals that are related to humans, focusing again on their CFTR proteins. So here, in just a second, we'll see some of the motifs found in the CFTR proteins from related animal species. So we can see that there's some transporter motifs, and we can see a few other motifs that are found in the CFTR proteins of other animals. Now let's go back and look at a few other options in the Blastweb view. When we scroll down, we can see a long listing of the protein sequences for animals. You can click on any of those links and it'll take you to the sequence that you can download and use in future projects. And you can scroll through here and see that there's a long list of data that's returned through that query. Now let's look at a few other views. So we'll click there and we can see a taxonomy view that shows the relatedness to other known animals with their genomes that have been sequenced. So we can see here, of course, as you'd expect, that the human CFTR is most closely related to other primates. As you scroll down, you can see a wide variety of other representative animals that have also been sequenced. So one more view that is nice to look at is the tree view. So let's select that, and you'll see in just a second that it displays a cladogram. And in this particular view, we can see the relatedness to other animals, again using CFTR as the only sequence that's determining the tree. Let's look at a few other options for how to view this tree. So if we change this to a slanted cladogram, we can see this view, which is a very nice and tidy tree view. The links of any branch represent the genetic distance or the evolutionary distance between the species. There are a few other views that we can look at. We can look at the radial view, and we can also look at the circular view. So you can see each of those presents the genetic relatedness in a slightly different manner. But let's return to the slanted cladogram because that's a very nice tidy view. So in this session, we've learned how to use Blastweb to compare and identify related protein sequences from other species.